Hello guys, well done for logging on and signing up to doing this lesson with us. Today's lesson is fundamentally going to be about revision. We're going to be doing a lot of revision. We're going to be covering the 10 topics that we've covered during lockdown, and this is in preparation for a short test that you guys are going to be doing next week. So we thought we'd do this lesson just to cover very, very quickly the 10 topics that you guys have been covering during lockdown. Before we get on to that, guys, what you're going to need for today's lesson is a pen, paper, pen or pencil, piece of paper or a notebook. So if you haven't got those with you to hand, pause the video now, run off and grab all the bits and pieces you need, get yourself settled down and get yourself ready for some learning. Okay, then hopefully everyone's back. Everyone's got all the materials they need to carry on with the lesson. We're going to get cracking on. The first thing we're going to be doing today, though, is a quick recap over last week's lesson, which was about the heart. So we've got 5 questions now that I'd like you to have a go at. Pause the video now and we'll quickly run through these questions afterwards. Okay, guys, well done for having a go at that. So we've got 5 questions now surrounding the heart and the blood vessels supplying it. The first question is, what blood vessels carry deoxygenated blood? Now, predominantly that's the veins, although if you remember that the pulmonary artery is the only artery that also carries deoxygenated blood, as it takes the blood from the heart to the lungs to become oxygenated. What adaptation does the left ventricle have to support its function? It has thick muscular walls. They're really, really quite important, guys. Thick muscular walls that allow it to produce that massive pressure so it can force the blood around our entire bodies. What ensures that blood flows in the correct direction around the heart? They were the tricuspid and bicuspid valves. If you put valves down for that, I'm more than happy, guys. Give yourself a tick if you put valves, or more accurately, if you put the tricuspid and bicuspid valves. Can you write the formula to calculate stroke volume? Well, I put the triangle there for you guys as a little bit of support for you. You should be able to do this. If we're trying to work out stroke volume, remember we just cover up the stroke volume, and then we know that stroke volume is equal to the cardiac output divided by the heart rate. Now, Sally has a stroke volume of 86 centimeters cubed and a heart rate of 60 beats per minute. What is her cardiac output? Now, I didn't say you needed a calculator, guys. I've kind of kept the numbers quite simple for this one. But if you, we know that it's cardiac output, so we cover up cardiac output, and then it's the stroke volume times the heart rate, or the heart rate times the stroke volume. And if you put the right words into this part of the equation, but you didn't have a calculator to do the actual sum, then you can give yourself a mark for that. But if you did have a calculator and you were able to do the maths, then you'll see that the cardiac output is 4,800 cubic centimeters per minute. And that unit's really important, guys. I know I bang on about this a lot, but really, honestly, a number in science has very little value if it doesn't have a unit attached to it. So give yourself a mark out of 5. Well done for having a go at those, guys. And we're going to move on to the rest of the lesson. So if you can remember way back when lockdown began, the first lesson that you had on lockdown learning was about cells. I realised that my ugly visage is covering half the image that I was hoping to show you to demonstrate a plant cell, but I can't do much about that now, unfortunately. So when you were looking at cells, there were 2 main types of cells that were considered during that lesson. They were prokaryotic cells, or prokaryotes, and eukaryotic cells. Now, the key distinction, if you can remember, for those 2 types of cells is that a prokaryotic cell has no nucleus, and a eukaryotic cell has a nucleus. Now, a bacterial cell is an example of a prokaryotic cell, and animal and plant cells are examples of eukaryotic cells. Now, we can remember the organelles. One of the ways that we can define them, firstly, we need to look at the organelles that are just found in an animal cell. On the diagram on the screen in front of you are all labelled in black. So that's the cell membrane, ribosomes, nucleus, cytoplasm, and mitochondria. And they're all found in an animal cell. But they're also all found in a plant cell. Plant cell has all of those, but it's got a couple of other ones as well. We've got the cell wall for structure and support. We've got the vacuole, which is used as a sugary store, a store for nutrients, as well as for structure and support. And we've got those really all-important chloroplasts. That's the area where the site of photosynthesis, where plants are able to produce their own energy. And then we move down to the bacterial cell. This is a prokaryotic cell, remember guys? So the first thing we can notice is that it doesn't have a nucleus. It has this chromosomal DNA all tied up in a bundle in the middle of the cell. They also have plasmid DNA. These are rings of DNA that float around in the cytoplasm of the cell. That points out the fact that bacterial cells, like animal cells and plant cells, have cytoplasm. They also have a cell membrane. That's very important. And they also have ribosomes because they still need to produce and synthesize proteins. Like the plant cell, a bacterial cell will also have a cell wall. That's more for protection and also for structure and support. Now, some of the things that a bacterial cell has that the plant or animal cell or the eukaryotic cells do not have, the first one is a flagellum. Now, this is the tail item to what looks like a bit of a tail at the bottom of the bacterial cell. It's used for locomotion. And they also have what's known as a slime capsule. Now, this is not all of them have this, but this is a means that the bacteria uses to protect itself from any harmful elements in the environment that it's in. So that's a very quick run through of the different organelles that these three different types, or the two main different types of cells have. And what we're going to do now, just to make sure you've got that all finalized in your mind, what I'd like you to do, guys, is to copy this table into your notes. And then I would like you to fit each of the organelles into each of the columns that is appropriate for them. It is worth mentioning at this point that some of those organelles will be found across more than one of the columns. So pause the video now, guys, and then have a quick go at dropping those organelles into the different columns. Okay, well done for having a go at that, guys. Okay, so I'm going to quickly span up the answers there. So as we can see, as you might be able to remember, that plant cells, animal cells, and bacterial cells all have a cell membrane controlling what goes in and out of the cell. They all have cytoplasm, an area for reactions to take place, and they all have ribosomes to produce proteins. But there it differs. The animal and the plant cell also has a nucleus, whereas the prokaryote, the bacterial cell, has chromosomal DNA. They don't have their DNA enclosed in a nuclear envelope, and they also have plasmid DNA, and these small rings of DNA that are free floating within the cytoplasm of the cell. Both plant cells and bacterial cells have a cell wall. Plant cells and animal cells have mitochondria. Plant cells also have the vacuole, chloroplast. And if you can remember, those two key distinctive organelles for bacterial cells was the capsule or slime capsule and flagellum. Okay, so well done for having a go at that, guys. Give yourselves a mark. Pause the video now if you've got any wrong and you want to adjust your notes, because this is a revision lesson, so it's important that you've got the correct information in your notes. Okay, the next lesson after we did cells, if you can remember, was about enzymes. Now, if you can remember all the way back to then, we remember that enzymes are proteins. They're made of protein, and they function as biological catalysts. And what that means is they can facilitate a reaction taking place. They can reduce the amount of energy required for an action to take place, or they can increase the speed at which a reaction takes place. And what they do is they have an active site. Now, that active site is a very specific shape, and that shape will only allow very specific substrates to be bound with the enzyme. And that very specific shape that just allows one substrate to be bound to them is known as the lock and key system. Now, when the substrate binds to the enzyme, it becomes known as the enzyme-substrate complex. And it's during that period of time that the enzyme's doing its job. Now, after it's done its job and the products are released, the enzyme itself is completely unchanged and can then go again to take part in another reaction where it will be either splitting or combining something. Now, one thing that's quite helpful is that many enzymes, when you're reading through bits of text or questions in your exams, many enzymes will end in with the last part of their word being an "ase," and that is just a kind of not hard and fast rule, but it's a bit of a rule of thumb throughout. Most enzymes you come across will end in with the last part of their word will end as "ase." Okay, so the limiting factors, again, same lesson. Because enzymes are made of protein, at high temperatures or extreme pHs, extreme environmental conditions, proteins can become denatured. Now, what that means is that they lose their structure and their shape changes as they become denatured. And as they change their shape, the active site, that key part of the enzyme's shape, also changes. And that means that the substrate doesn't fit in that active site anymore. So the lock and key system no longer works, and the enzyme cannot bind with the substrate. Now, we've got on the screen in front of you, we've got 3 different graphs. What I would like you to do is to pause this video now, copy the 3 graphs into your notes, and then label what is occurring at each of the, or write down what is occurring at each of the labeled sections of the graphs. Pause the video now, guys, copy them down, and then comment on each labeled section. And I'll be on hand afterwards, and we'll go through what I consider to be the right answers for those. Well done, guys. Okay, so we're back in the room. Okay, so we're looking at the temperature graph here. Now, as temperature increases, if you remember back to year 7, year 8 particle chemistry, as some substances get more energy and more heat, they get more energy, they move around more. So the increasing temperature causes the enzyme and substrate to move more, and this increases the chance of them coming into contact with one another, and this will increase the rate of reaction. Now, enzymes will function best at a very specific temperature. At this temperature, the rate of reaction is at its fastest, and it's known as the optimum temperature. That's a key word there, optimum, guys. It means it's functioning at its best. Now, when you get to really high temperatures, the enzyme will denature. Remember I said that means it changes its shape. The enzyme can no longer bind with the substrate, and this means the rate of reaction will slow down very, very quickly after this point. So that's temperature, guys. Well done if you got that right. Pause the video now if you didn't, and make any changes to your answer, because as I say, this is revision notes, and you do need these to aid you in your test next week. Okay, so moving on, pH. Now remember I said that extreme pHs, the protein will denature. So at low pHs, in strong acidic conditions, the enzyme denatures, it changes its shape, and much as we were seeing with the temperature, it means that the substrate can no longer bind with the enzyme, and the rate of the reaction is decreased, is slowed down. Much like with temperature, enzymes have an optimum pH at which they function. And again, that's what's occurring at section B of this graph, the top of the graph. The rate of reaction is at its fastest. It's at a very specific pH, and you'll see there's a sharp climb to it and then a sharp drop off after it. So see the sharp drop off. This is at a high pH, so it's a very alkaline solution. And again, that will still cause the enzyme to denature, change its shape. And as we probably guessed, guys, it can no longer bind with the substrate, and the rate of reaction reduces. So that's pH. Again, pause the video, make sure your notes are correct. These are really important for your revision, guys. And now the last one we're going to look at, the last graph we're going to look at, is the substrate concentration. Now, as we increase the substrate concentration, it increases the chance that the substrate will find the enzyme, will come into contact with the enzyme, and thusly the active site and the reaction can occur. So as you're increasing the substrate concentration, the rate of reaction will increase very, very quickly until you reach point B. Now at point B, the concentration of the substrate becomes high enough that all of the enzyme in your mixing pot in your area is being used. And after that point, it doesn't matter how much substrate you throw into that mix. If all the enzymes being used, the rate of reaction cannot get any faster. And so that's what you can see it be there. That's why that long line continues like that, because all of the enzyme is being used at this point, and any further increase has no impact on the rate of reaction. Okay, the next lesson you was looking at some of the digestive enzymes that our bodies produce to enable us to break down our food into small enough parts so that we can get it into our system and actually into our bloodstream and around to all the different cells that we need. Now, digestion actually begins as soon as you put food in your mouth and you start chewing it. That's known as mechanical digestion. You're breaking the food up, breaking it into smaller pieces, but it doesn't matter how long you chew your food for, guys. You're never going to break it up into a small enough part just with your teeth to be able to get it into your in through your gut wall, into your bloodstream, and into the cells that you need it. So we use digestive enzymes to break those products down even smaller so that they can either diffuse or through active transport be transported through the gut wall into our bloodstream and circulated to our bodies. The first enzyme that we will come across, it occurs in our mouth. It's a part of our saliva, and it's known as amylase. It's a type of carbohydrate, and that's because it breaks down carbohydrates. It breaks down starch, which is a complex carbohydrate. You can see this chain of hexagons here. That's starch. That's a chain of glucose molecules. And amylase breaks that down into individual glucose molecules, which can then be transferred through our gut wall into our blood system with bioactive transport. So the next, as the food travels down through our esophagus, it reaches our stomach, and in our stomach we get another enzyme. This is known as protease, and that breaks down proteins. It breaks them down into the base units of amino acids, and again, they're small enough to be able to be transported through our gut wall or diffused through our gut wall into our bloodstream. Now, finally, as our food leaves our stomach and enters our small intestine, our pancreas will secrete a load of lipase. Now, lipase is the special enzyme that breaks down fats, and it breaks them down into glycerol and fatty acids. But our pancreas doesn't just secrete lipase, it also secretes amylase, the carbohydrates, to break down carbohydrates, and proteases to break down proteins. So whilst in our mouth we have amylase breaking down carbohydrates, we have protease breaking down proteins in our stomach. In our small intestines, we have lipase breaking down lipids, but we also have the enzymes to break down carbohydrates and proteins. So all three occur within our small intestine, and they're all secreted by the pancreas. So guys, we're going through this at a race of knots, I am aware, but what I'd like you to do now is to copy this diagram into your books. Pause the video, copy the diagram down into your books. We've got 3 key organs that we've just been talking about identified on this diagram, and what I would like you to do is to label this diagram in your notes. I'd like you to include the organ, the digestive enzyme, and both the substrate and the reactants that are produced by that reaction. So copy those down, copy the diagram, put those onto your diagram, pause the video, and we'll be back on hand to go through the answers in a few minutes. Well done, guys. Well done for having a go at that. We're going to just quickly run through these answers. The first one of which we can see is in our mouths, we have salivary glands. They release amylase. That breaks down starch into simple sugars. Then in our stomach, we had protease. Now that's an enzyme that breaks down proteins, and it breaks them down into amino acids. And then finally, we have our small intestine secreted by the pancreas. And in our small intestine, we will find lipases to break down lipids, amylase to break down starch, and we'll find protease to break down proteins. So we get all 3, as I said before, we get all 3 found within the small intestine, all 3 groups of digestive enzyme. Okay, well done for having a go at that, guys. You're doing really well. We're running through this at quite a rate of knots. So we're going to move on to the next topic that you guys looked at, and that was diffusion. Now, diffusion is the net passive movement of particles from a region of high concentration to regions of a lower concentration. Now, if you can pause the video very quickly and just copy that statement down into your notes. I know you did so during the lesson, but it's such a key statement, guys, that I really do want to make sure that you've got it in your minds recently so that you can, it'll be at hand in your minds when you need it for next week's exam. Now, what we can see is from the 2 diagrams that we've got here, we'll go for that statement first. Firstly, diffusion is the net passive movement of particles. Because it's passive, it doesn't require energy. That's a key factor for diffusion, guys. There's no energy requirement for it at all, and it occurs from a high concentration to regions of lower concentration. So it's a movement down the concentration gradient. Now, the 2 diagrams that we've got here show you that the one at the top here shows you diffusion occurring in a beaker with some water molecules, and you can see here the concentration is quite high, and then it just slowly spreads out until they're evenly distributed and they're at equilibrium across the entire solution. And we've got simple diffusion down here, and the reason I put these 2 images on, guys, is to really show you that diffusion can occur with no membrane, but also as long as the particles are small enough to be able to fit through a membrane, then actually diffusion diffusion will occur across a membrane as well. It doesn't need a membrane for diffusion to occur, but it can occur across a membrane. One example of that would be when we diffuse oxygen out of our lungs into our bloodstream, or out of our bloodstream into the different cells in our body. So we have 4 ways of being able to improve the rate of diffusion. So a larger concentration gradient, a higher temperature, a larger surface area, or a shorter diffusion pathway. All of those facets, all of those facts can work to increase the rate of diffusion. And a lot of the various places where diffusion occurs in our bodies, you will find one, if not more, of these kind of special adaptations to improve the rate of diffusion. The next lesson you guys looked at was osmosis. Now, again, we've got another passage here. Osmosis is the net passive movement of water molecules through a semi-permeable membrane from a region of high water concentration to a region of low water concentration. Now, again, guys, pause the video quickly, copy that statement down. I know, again, you will have copied something very similar into your notes when you initially did this lesson, but have another go at it to make sure it's clear in your mind and you know that statement. And going through that statement again, we see the word passive, so we know that there's no energy required because it's a passive movement. We also see again that it's going from a high water concentration to a low water concentration, so it's down the water concentration gradient. But this one, unlike diffusion, does require a semi-permeable membrane, and all that means is it needs a membrane that has some small openings in it that's small enough for water to pass through, but also too small for larger molecules to pass the other way. And that will cause the this area here where there's a large concentration of water and a lower, these blue dots of water molecules, lower concentration there, so the water will move from this side into that side. Okay, so that's osmosis, guys. The last mode of transport that you guys did, it was over a few lessons, was active transport. Now, again, we've got a red box, so pause the video here, guys. Write that down. Make sure you've got it in your notes because it is a key statement that I do want to make sure that you guys understand. Now, active transport, as it says, I'll let you pause the video and we'll go through it now. So active transport is the movement of molecules across a membrane from an area of low concentration to an area of high concentration. Active transport often occurs through special proteins and always requires energy. So we break that down like we did the last few statements. It needs energy. It always needs energy because you're moving things against the concentration gradient. It also has to happen across a membrane. So there needs to be a membrane for active transport to work. And finally, it moves from a low concentration to a high concentration. So unlike osmosis and diffusion, it works against the concentration gradient. So what I'd like you guys to do now is to draw 3 large circles, much like you can see in the middle of the screen now. Label them diffusion, osmosis, and active transport. It's a bit of a Venn diagram. And what I would like you to do is to input the various statements that you can see around the screen into the correct part of the Venn diagram. Pause the video now, guys. Have a go at doing that into your notes, and I'll be on hand when you've finished to go through the answers. Well done, guys. Well done for having a go at this. We're rattling through this at quite a rate of knots. Okay, so we'll start with the stuff in the middle of the 3 that is active in all 3 of the forms of transport. It's the movement of particles. All 3 of them involve that. And all 3 of them occur in nature. Diffusion occurs in our lungs. Osmosis will occur in our kidneys. It will also occur in the in plant cells to maintain their structure. And then active transport occurs in the roots of plants to absorb the minerals into the root hairs. Okay, so that's the occurring in nature. So it occurs from a high to low concentration. We know that was both diffusion and osmosis, but not in active transport. Active transport and diffusion both involve the transport of solutes. Osmosis and active transport both need a membrane of some description to function against. Diffusion and osmosis were both passive movements, so they require no energy. Diffusion is how carbon dioxide will get into a leaf. Osmosis involves the movement of water only. It's how water keeps plant cells turgid and how plants maintain their structure. Active transport is how minerals get into root hair cells, I mentioned just briefly earlier on, and it works against the concentration gradient. So well done if you've had a go at those, guys. Oh, sorry, one more. It also, active transport, if we remember, requires energy. Well done for having a go at that. If you've got any in the wrong places, pause the video now very quickly and just make sure you've got them in the right places for your notes. Because as I say, this is really going to be very useful for you for revision for the test that we're going to be doing next week. Okay, so the next lesson that you guys looked at was about respiration. Now we've got a red box. It's slightly different from the one you will have copied down in your lesson. Can you pause the video now? Just copy those words down into your notes. Respiration is the process of releasing energy from nutrients. Respiration is not breathing. The amount of exams I've marked where people have made that mistake, respiration is not breathing. If you ever mistakenly put the word breathing instead of respiration in an exam answer, you will get no marks, guys. So please, I've made it bold, I've capitalized it. Respiration is not breathing. Now, aerobic respiration, which is the type of respiration that we as humans mostly use, occurs in the mitochondria of our cells, and it involves reacting the oxygen that we're breathing in with the glucose from the food that we eat. And that gives us this equation. Glucose plus oxygen is equal to carbon dioxide, or it creates or converts to carbon dioxide and water, and it releases energy. We use that energy for a whole range of reasons. For everything we do, fundamentally, that is our only source of energy, and we use it for moving. We use it for keeping warm, and we use it for growing. We use it for everything that we do. So looking at this equation, we can see that the reactants are glucose and oxygen. The products are carbon dioxide and water, sometimes referred to as waste products. And because the reaction produces energy, we can say that the reaction is exothermic. It's releasing energy. And this reaction actually releases quite a lot of energy, 38 ATP. So we're moving on. All the while our body's doing normal stuff, we are able to produce enough energy, we can get enough oxygen into the different cells of our body for aerobic respiration to continue. You can see this guy sitting down on his settee there. He's not using a lot of energy, so his muscles and the various parts of his body are able to use purely the oxygen that they're getting into their body is enough for their cells to produce energy. When we start to exercise strenuously, the amount of oxygen that's getting to our muscles and the different parts of our body starts to not really be enough. And when the circulation system can't supply enough oxygen to our muscles will then switch into anaerobic respiration. And anaerobic respiration is the incomplete breakdown of glucose, and it doesn't require oxygen. And that's why it's called anaerobic, because it's no oxygen respiration. Now, anaerobic respiration isn't ideal, guys. It releases far less energy than aerobic respiration. And we can have a look at the equation now. We get glucose. There's nothing else that's added to it. It's just broken down within the cytoplasm of our cells, becomes lactic acid, and it releases just 2 ATP from that entire molecule of glucose. Now, so we've got glucose is our reactant. Lactic acid is our product or our waste product. And again, although it's only releasing a little bit of energy, it is still releasing some energy. So it's still an exothermic reaction. Now, you will be aware of lactic acid. If you've ever done any exercises and you felt your muscles burning, that hot sensation in your muscles when you're exercising is simply the buildup of lactic acid. And it goes away once you start resting and your body can revert back into aerobic respiration. Okay, guys, so we've just got a simple 5 questions here, or 6 questions. I've got an A and B, and question 5, I cheated a little bit. We've got 6 questions, guys. So just pause the video at this point, run through these questions, and I'll be on hand afterwards, and we'll be going through the answers. Okay, guys, well done. Well done for having a go at those questions. Okay, we're getting there. We're going along at a cracking rate here, guys. So what was the word equation for aerobic respiration? A couple of slides ago and a good few weeks ago now, the word equation for aerobic respiration, if we can remember. Oh, sorry. I've gone the wrong way. The word equation for aerobic respiration is glucose plus oxygen. There should be an arrow here. Just imagine there's an arrow here. It can be converted into carbon dioxide and water and energy. Okay, so what is the product of anaerobic respiration? Remember, that was lactic acid. That's the burning sensation we get in our muscles under heavy exercise. Under normal conditions, our bodies use aerobic respiration. Why? Well, if you can remember about the amount of energy that was released for every individual glucose molecule, we'll understand that aerobic respiration is far, far more efficient. It produces far more energy for every glucose molecule, 38 ATP compared to just 2 when you're doing anaerobic respiration. So it's more efficient. That's why our bodies normally use it. What are the reactants of aerobic respiration? Well, we've already written the word equation down, so hopefully, guys, you can work out from that what the reactants were, but they were glucose and oxygen. Now, this is a kind of connection question. This is connecting the previous set of lessons to what's occurring in this lesson. We're going to, by what process do our cells use to uptake oxygen from the bloodstream? Now, oxygen is a tiny, tiny molecule, and our bodies are able to uptake the oxygen from the bloodstream simply by diffusion. Now, what process do our cells use to uptake glucose from the bloodstream? Glucose is a much larger molecule, and it cannot simply diffuse through the membrane. It's not water, so we can't use osmosis to transport it across. Therefore, we have to come to the last, and you use active transport to transport oxygen out of our bloodstream and into the cells that require the glucose. So well done for having a go at those questions, guys. Give yourself a mark out of 6 if you got them right. Well done for having a go. So we're going to move on to the next lesson. The next lesson was only a couple of weeks ago, so we're getting close to the end now, guys. It was all about the blood and the different types of blood vessels and components of blood. Now, if you can remember, the largest part of our blood was made up of plasma. And this was used to carry dissolved nutrients and waste products and oxygen and things like dissolved nutrients and waste products around our body to all the cells that require them and to remove the waste products. We had erythrocytes. They were the next largest component of our blood. That made up 45% of our blood, and their main role was to carry oxygen. They had that biconcave shape. They had no nucleus to increase their surface area so they could really get lots of oxygen into the cell. Phagocytes, they were a white blood cell, and they were the white blood cells that engulfed and digested their pathogens, and they had lots of digestive enzymes within the cell body to enable them to do that. Then we had lymphocytes. Lymphocytes were, again, a type of white blood cell, and they're used to identify and deactivate pathogens. And they do that by having those tiny, very specialized hooks that I told you about. Remember the antibodies that attach to these certain specific parts of a pathogen? And then we had platelets. These were the tiny cell fragments, sticky cell fragments that can bind together and form a scab over a wound site to prevent us losing any blood. Now, the vessels, we had 3 different vessels that we went over during that lesson. One of those vessels was arteries, and remember their main function was to carry blood away from the heart. And their adaptations were that they had a very thick elasticated wall to withstand that pressure, and they also had that small lumen to maintain that pressure. Now, veins carry blood to the heart. Now, by the time the blood's got to the veins, it's at a much lower pressure, so they didn't need the thick elasticated wall, so their walls are much thinner and unelasticated, and they've got a much, much larger lumen to reduce the drag as the blood flows through the blood vessel. And remember, they also had those very special valves to ensure that the blood doesn't all end up collecting down at our feet and it doesn't go the wrong way down the vein. And the last of the blood vessels we looked at was those small capillaries, the business end of the blood system. These are tiny, tiny little blood vessels, and this is where the oxygen and the nutrients are diffused or transported out of the blood system to the cells that require it, and where the blood collects all the waste products before taking them to the right parts of our bodies to be disposed of. So guys, on the back of that, what I'm going to get you to do is just a very quickly, or pause the video, or copy and complete this table down. So in a minute, pause the video, guys, copy this table down. I'd like you to write the function for each of the components of the circulation system, and then give me an adaptation for that function. So pause the video now, and I'll be back on hand to go through the answers. Okay, well done for having a go at that, guys. We will go for the answers now. So if we can remember, plasma, its function was to carry those dissolved nutrients and waste products in the circulation system, and it has no real special adaptation. Now, erythrocytes, these were the red blood cells, carry oxygen. They have no nucleus and a biconcave shape. That's that special pinched in the middle. I will also accept if you put into there that contains hemoglobin. That was the special protein inside the red blood cell that holds on to the oxygen. Now, we've also got phagocytes. These were the white blood cells. Their function was to engulf and digest pathogens, and they do that by having lots of digestive enzymes within their body, within the cell body. And then we've got the lymphocytes. These are, again, another type of white blood cell. They identify and deactivate pathogens, and they do that by having lots of antibodies sticking out from the side, outside of their set of the cells. And then we had platelets. These form scabs over wound sites to prevent blood loss, and they're basically made of sticky cell fragments that combine together to block that opening. Now, our arteries carry blood away from the heart. That's the key bit there, away from the heart. They have thick elasticated walls to withstand that pressure. I haven't put it in here, but they also have a very small lumen to maintain that pressure. Any of those adaptations, or both of those adaptations, are more than fine for this as an answer. We move on to veins. They carry blood to our heart, and they have thin unelasticated walls because the pressure's gone away from the blood system by this point. The blood's done its job, it's traveled around the body, the pressure's fallen, and they don't need those thick elasticated walls of the arteries. What they do need is a large lumen. Again, I've missed that one out of this statement as well. And they also have those valves to stop the blood traveling in the wrong direction through our veins. And then our capillaries, this is the site of nutrient and oxygen and waste product exchange. And if we can remember, they're very, very small. Only one red blood cell can fit through at a time. They have a large surface area because they are so small. And they also have very thin walls. Their walls are only one cell thick. So that's that short diffusion pathway. These are the ways that we can increase the rate of diffusion. Okay, guys, well done for doing that. We're going to move on to the heart. I've clicked the button already, and we're going to go through a very quick whistle-stop tour of the heart, the route that the blood travels through our heart. If you can remember back from last week, we leave the body, all of the veins from our body collect in our vena cava. That's the main vein that supplies the blood back to the heart. It enters the heart at the right atrium. Remember, it's on the left-hand side of the image, but it's the right atrium because you're looking at a mirror image. That's how the heart would appear in someone's chest in front of you. Then the blood goes through those tricuspid valves into the right ventricle, and then out of the right ventricle in the pulmonary artery, still deoxygenated, carrying the blood to the lungs, where it gets oxygenated, and then it returns by the pulmonary vein into the left atrium. The left atrium, at the next contraction of the heart, the blood squirts back past the bicuspid valves that separate the left atrium and the left ventricle into the left ventricle. Then on the next contraction, the blood is squirted out of the left ventricle into the aorta to return to the rest of the body. And if we can remember, guys, that left ventricle had that really thick muscular wall to enable it to produce enough pressure to pump the blood all the way around the system, all the way around our bodies. Okay, guys, so having just had a quick recap on that, what I would like you to do now is, if possible, I would like you to copy and label this diagram of a human heart. Now, if you're drawing, if you're not confident with your drawing skills, you can always use the square one that we did on the last slide, and making sure you label the correct blood vessels into and out of the heart. But if you feel quite confident with your drawing, have a go at copying this diagram down, guys, and then labeling it for me. Pause the video now, do that, and I'll be on hand afterwards just to go through the answers. Okay, well done, guys. We're going to go through the answers now. So if we can remember, the blood collects in the main vein that feeds the heart, and that is the vena cava. So that's the main vein that supplies all the blood. It takes the blood into the right atrium. The blood then passes the tricuspid valve down here into the right ventricle. Then the next contraction forces the blood up this blood vessel into this blood vessel here, and that blood vessel is the pulmonary artery. The blood then goes to the lungs, loses its carbon dioxide, grabs a load of oxygen, comes back into the heart via the pulmonary vein. It enters the heart at the left atrium. It then goes past the bicuspid valves and enters the left ventricle, which has a very thick muscular wall. Remember, guys, that thick muscular wall, I've added it onto this diagram because it is such an important fact to be able to remember. That thick muscular wall, giving it enough pressure to squirt the blood into the aorta and supply that blood around the entire body. Well done for having a go at that, guys. Correct any answers that you've got wrong. Other than that, well done for having a go, guys. We're going to move on to the last slide of this lesson, and that's your homework slide. Now, I'm not going to set you guys anything to upload to Shobi this week, because next week we're going to be having this test over the subjects we've covered during lockdown. And what I would like you to do is, instead of giving you an exercise to do, what I'd like you to do is to do some revision over the 10 topics that we've covered, the 10 topics that I've just covered in this lesson. You can use either the previous lessons that were uploaded by Miss Page or myself, or you can use this lesson, go through the subjects, make some spider diagrams, some flashcards, whatever technique you use, but spend some time going over these topics in preparation for next week's lesson. Well done for sticking with me this long, guys. You've done really well this week. It's an awful lot of information I've tried to cram into your brains in a very short period of time, but well done for sticking with me. And I look forward, I hope you all have a lovely week. Hopefully the sun's going to be shining for us a bit more this week. Have a lovely week, and I look forward to seeing you next week, guys. Have a great one. Take care and all the best. Cheers now. Bye-bye.